Okay, welcome back once more. So in this video, I want to discuss the mechanism or the process of translation. So just like these other processes, starting from DNA replication, transcription, up to this point, translation, they are all subdivided into three. There are three stages which are involved here. So there's what is known as initiation. Then the second step is what is known as elongation. Then the third step is termination, where the process comes to an end. So initiation, when it comes to translation, this is where a lot of activities do take place. Then when it comes to elongation, this is where now just the synthesis of a polypeptide chain is growing. That's elongation. Then termination, the time at which this process of translation now comes to an end. So this is what these are the three stages in which this process of translation is divided into. So we want to understand what happens during initiation, then also what happens elongation, then also what ends the process, which will be termination. So as I've already mentioned, a lot of activities here will take place during the process of initiation, and this is what we want to look at. So before we get into this, let's first define and understand what do we all mean by this process of translation. So translation is also termed as protein synthesis. Protein synthesis. Okay, so this is so protein synthesis, that is what we call translation. So when you talk of translation, this is just the process by which proteins are produced from the mRNA sequences. And we know that these proteins will be in a form of amino acids. As we have already discussed about the properties of the genetic code in our previous video. If you have not watched that video, you can go and watch so that you learn more about the properties of genetic code, which do explain more also about these amino acids that are the building blocks of proteins. So proteins are produced from the RNA-based sequences. Such a process is what is known as translation. And we have to understand that this process of translation takes place in the cytoplasm of a cell. So be it prokaryotic cell or eukaryotic cell, translation takes place in the cytoplasm. The reason being why it takes place in the cytoplasm is because it is in the cytoplasm where we have ribosomes. So these ribosomes, we know the function of ribosomes. It is to produce or protein synthesis. Even from high school, the function of ribosomes has not changed. It was still for protein synthesis. So that mRNA has to interact with these ribosomes so that it can finally produce proteins, which is the goal or the central dogma of molecular biology. So that is what happens, being prokaryotic cell or eukaryotic cell. So now, how does this process of translation start? Initiation. We said that a lot of activities do happen in this stage, at this stage of initiation. So what happens here? So the first process that we have to understand is aminoacylation. So we have to understand the process of aminoacylation. This is the first of all. Before this, we know that we only have 20 amino acids which do exist in nature. There are only 20 amino acids which produce millions of proteins that exist in living organisms. Now these 20 amino acids are always found in the cytoplasm of the cell and in their inactive form. So meaning that if the ribosomes do read a sequence of mRNA, a triplet code of that sequence of mRNA that is going to send a signal for a particular amino acid to be picked to come to the ribosomes. So meaning that one codon will be read, then one amino acid has to be picked from these 20 which are in the cytoplasm. So we are saying here that they exist in their inactive form. So they have to be become active. Once they become active, then we also have to look for an alternative which can transport these amino acids from the cytoplasm and take them all the way to the ribosomes for assemblage and formation of polypeptide chain, which is finally going to give us proteins at the end. So this is what happens. So we are going to look at how these amino acids become active, then how they are transported also from the cytoplasm to the ribosomes. It is ribosomes are found within the cytoplasm, but we mean like picking these amino acids from the cytoplasm and take them to an organ, a special organelle within the same cytoplasm, which are now ribosomes. So this process of aminoacylation, it is the most important that we have to understand about the activation of the tRNA as well as the activation of amino acid charging and all of those. So aminoacylation, this is the process of charging tRNA. This is the process of charging tRNA. So tRNA, transfer RNA, this is the molecule which is capable of transporting these amino acids from the cytoplasm to the ribosomes. That is only if that amino acid becomes active. So when a particular amino acid becomes active, it will now have to attach itself to an appropriate tRNA. Then that tRNA, as you can see here, this is the tRNA. Then this tRNA now can transport that amino acid to the cytoplasm. So charging of this tRNA, that is what is known as aminoacylation. Now this process of aminoacylation involves two stages. The process of aminoacylation involves two stages. So the first stage is what is known as activation. Activation of amino acids. And amino acids. So this amino acid has to be a target amino acid. Activation of an amino acid. So we want to see how these amino acids are activated. Maybe before we explain that, the second step during the aminoacylation is what is now known as charging. Charging now here involves the attachment of an amino acid to the tRNA. So this now involves the attachment of that an amino acid to the tRNA. That's what is known as charging. So these two are what comply the process of aminoacylation. So let's go back and look at how this activation of an amino acid takes place and what is involved there. Then we can come to explain about this charging of amino acids. Then we can say we are done with aminoacylation. So what happens here now? So activation of amino acid, this process is catalyzed by an enzyme. Okay, this process is catalyzed by an enzyme, amino acid tRNA synthetase. So that is an enzyme, amino acid tRNA synthetase, or synthetases in plural. Okay. So these are enzymes which catalyze this process of activation of an amino acid, which is the first stage during aminoacylation. So what really happens here? So this the activation of amino acids involves the formation of amino acid AMP, or amino acid adenosine. So we are saying that activation of amino acid, which is the first stage during the process of amino acylation, this one involves the formation of amino acyl adenosine or amino acyl AMP. So that one is also what we call amino acyl adenosine. Okay. That is okay, like this. So the process of activation of an amino acid involves the formation of this molecule, but this is only becomes possible due to hydrolysis or breaking down of ATP. So to hydrolysis, or you can say breaking down of ATP. So we know that energy in a cell, it is in a form of ATP. So activation is nothing but just providing an ATP energy to that target amino acids which a tRNA wants to pick. So this is what happens. This molecule is going to be produced. Amino acyl adenosine is what is going to be produced. So an equation in this case is okay. An equation in this case is we had this amino acid which was uncharged, so we have an amino acid which is uncharged. This amino acid will react with ATP. So this ATP is what now is going to be broken down or hydrolyzed. So when it is broken down, we know that it's not now going to give us what? This is now what is going to form that amino acyl adenosine, which we call amino acyl. Let me use this color. Okay, so this is now what is going to give us that molecule amino acyl AMP or amino acyl adenosine. AMP, this one will be plus an inorganic molecule is going to be released. So this inorganic molecule is now going to be what we call the pyrophosphate. So this is going to be amino acid plus pyrophosphate, which is abbreviated as PP prime. PP prime, like that. So this is an inorganic molecule which is now going to be released. But our target in this case now, it was that amino acid AMP. So we say that this reaction is what is catalyzed by a set of enzymes known as amino acid tRNA synthetase. So that's what we have there. Amino acid tRNA synthetase. This is the enzyme which catalyzes this reaction. tRNA synthetase. Like this. So when this happens, then now the amino acid is said to become charged. So our goal in this case was to produce this molecule, amino acid AMP. Then it is this now molecule, amino acid AMP, which is going to form a bond with the ATP. It's now going to attach itself to the one of the tRNAs, which is its appropriate tRNA. That is it. This is all about activation of an amino acid as the first step in amino acylation. Then from here now, we can talk about the second step of amino acylation, which is we said is charging the attachment of an amino acid to the tRNA. So in this case now, that target amino acid has become active. It has now become active. We can get rid of it. Okay. Then now the second step, we said the second step in the amino acylation is what is known as charging. So what happens here in charging? Charging now involves the attachment of these activated amino acids to this tRNA. So how is that done? How is it done? In this case now, that amino acid AMP or amino acid adenosine is going to, so we have this in this case now, that amino acid AMP will now react with the tRNA. It will react with the tRNA. Then at the end, what are we going to have? At the end, what we are going to have here, we are going to have amino acid tRNA. So this molecule AMP is going to be released from the amino acid. This molecule is going to be released where its place is going to be taken by that tRNA. So we are going to have a molecule at this side which is known as amino acid tRNA. Amino acid tRNA here. Then this will be plus this molecule now, AMP. So in this case, what we see that an amino acid has formed a complex with the tRNA. That is what we mean by charging of an amino acid at the second step of this tRNA. Now, at this point, what has happened is that this amino acid AMP is going to attach itself to where? To the 3' end or that of its target tRNA or its appropriate tRNA. So in this case, if this is our tRNA, that's where this amino acid is going to attach itself here at the 3' end. So this is now going to be that charged amino acid. Now, let's also look at the characteristics of that 3' end of each and every tRNA. That makes it possible for this process of aminoacylation to take place. So one characteristic of the tRNA is that it has got these arms. It has got this arm, then this arm which is known as the D arm or D loop, this arm the L loop, then down here this is the codon loop. So these are what tRNA does contain. So let's focus our attention at the 3' end here. At the 3' end here, we can see this hydroxyl group. Hydroxyl group. You know the monomers of all nucleotides or nucleic acids, sorry, like DNA and RNA. So at the 3' end, there is always what? A hydroxyl group. So that is what we have there at the 3' end. So now, another interesting point is this one here. Each and every tRNA has got that, has got this base sequence at the 3' end, which is a CCA. CCA simply CCA can carry amino acids. CCA can carry amino acids for you to remember, for easy to remember, like that. Can carry then A for amino acids. So this is the codon which is always found. Amino acid. So CCA, this is a codon which is always found at the 3' end for each and every tRNA. So the reason why each and every tRNA has got this codon is because this codon plays an important role during the formation of a bond between an amino acid and this tRNA here. So it is a bond which it is a codon which makes sure that the process of aminoacylation takes place at the 3' end. That's why each and every tRNA will have to have this codon CCA at the 3' end. So let's see what happens. In this case now, we know the structure of amino acids. So let's say we have the structure of amino acid at the, here we have this. Then what do we have here? We have OH. Okay, something like this. Then this is what we have, and H2 here. So what will happen now is that this amino acid tRNA has got this molecule. Because that is an amino acid, so it has got this. So there is this terminal end, the carbon terminal end, which is for for carboxyl, the carboxyl terminal end. This carboxyl terminal end is the one now which forms a bond with the free hydroxyl group at the 3' end there. At the 3' end there. As a result, then we see that amino acid being attached at the 3' end, like that. So it is the C terminal end of that amino acid which attaches itself or forms a bond with the 3' end of the tRNA. So that's what happens. So each and every amino acid will have to undergo the process of aminoacylation, activation and charging, then attachment of that amino acid at the 3' end. Then when that amino acid attaches itself at the 3' end of this tRNA, this now tRNA is said to be charged. When an amino acid is carrying, when the tRNA is carrying an amino acid, it's said to be charged just because it's carrying an activated amino acid. So this is what happens during the during initiation. This is all about during that initiation step of both be it prokaryotic or eukaryotic cell. This is what happens. Now, from here now we can move on to look at the what will follow next after all these takes place. So understand aminoacylation, the activation and charging of those molecules. Then at this point now, the process of translation can start. So how does it start? So the process of translation now will start when we have this mRNA. Well, this mRNA, which now is in a form of messenger RNA. So we have messenger RNA carrying that genetic information in its form. So this messenger RNA contains bases here, base sequences here. So it has got base sequences. Now, this mRNA, when it is released from the DNA during where it was being transcribed, it has been released from DNA where it was being transcribed. If it is in eukaryotic cell, we know that it's going to undergo processing, modification, those splicing and the like. Then now, at this point, we see that it has been released into the cytoplasm. If we are talking of prokaryotic cell, this mRNA translation and transcription here takes place at the same time when it comes to prokaryotic cell, just because they all take place within the same cytoplasm. So what will happen at this point now is that this mRNA, when it has been released from that DNA, where it was being transcribed, and it has undergone the process of the processes of modification and the like, is now going to go straight to the ribosomes. So we know that the ribosomes are the organelles which are responsible for protein synthesis. That's where protein synthesis is going to take place. The reason why to have to go to the ribosomes is because these base sequences which it's carrying have to be read, and it is on the ribosomes which can read the sequences on this mRNA. As it reads, then it's going to interpret which amino acid is specified by which triplet code, codon, like that. So now we are saying that this will now go to the ribosomes. So now there are two types of ribosomes. There are two sets of ribosomes, be it in prokaryotic cell or eukaryotic cell. There's what we call large subunit, and there's also a small subunit. So prokaryotic cell, they have got a large subunit as well as small subunit. Also eukaryotic cell, just like that. So the large subunit, let's start with eukaryotic cell. Let's start with eukaryotic cells. So eukaryotic cell, the large subunit, the segmentation of the large subunit is 60S. So we are saying that the segmentation of the large subunit with regard to eukaryotic cell is 60S. S for segmentation. Then its small subunit is 40S. Then overall, the total segmentation is 80S. This is the total segmentation of eukaryotic cell. So we don't add 60 plus 40 to give us 100. No, if you have been asked about the total segmentation of Eukaryotic ribosome, it's a 80S, but each large subunit has got 60S, the small subunit has got 40S. Then now, what about prokaryotic cell? So when it comes to prokaryotic cell, the large subunit of prokaryotic cell, so in this case now, these are prokaryotic cells. So the large subunit of prokaryotic cell, it's a 50S. It's a 50S. S for segmentation, then the small subunit is the small subunit is the 30S. Then total segmentation of the ribosomes in prokaryotic cell, it's now 70S. So still more, we don't add 50 plus 30 to give us 80. It's that 70S. This is these are the segmentations of the ribosomes with regard to prokaryotic and eukaryotic cell. So what happens now? What happens now when this mRNA has been released to go to the ribosomes? It's going to go to the small subunit. So it's going to interact with the small subunit first. So if we're talking of prokaryotic cell here, meaning that this one is going to go straight to the small subunit, which is going to be the 30S. So let's say that is the small subunit. This is the small subunit. That's where this mRNA is going to attach itself. Once it binds to this small subunit of be it prokaryotic cell or eukaryotic cell. So if it is eukaryotic cell, then this we are saying that this is the 40S. If it is prokaryotic cell, that is the 30S. That's where this mRNA is first going to go to bind itself. When this happens, the large subunit is also now going to be communicated. So it will be communicated and come here and bind just like this to form this complex. So what we have at this point is that we have this, this is the small subunit, then this is now the large subunit at the top there, large subunit. This is what will happen. So this complex will now be formed. So what do we see next at this point? Firstly, this large subunit is divided into 3 different sites. So let's divide this into those 3 different sites. So it is divided into 3. So there's that one and this one. So these sites have got names. So this site here, far left, this is the E site. Then this one at the middle, this is the P site. This one far right, this is the S site. So these letters come from this S site. It is come from acetyl site. Then P site, peptidocyte. Then P. Then this E site, this is E for exit. So these are the 3 sites that the large subunit has. So want to see the significance of each and every site. The significance of each and every of these 3 different sites. So the P site and the like here. Then now the process of reading will start. So as we said, these bases on the mRNA, they are read in groups of 3. They are read in a triplet base. So 3 bases or triplet base. 3 bases will be read first. As we know that the genetic code is non-overlapping, so these bases, we expect them to be read sequentially. They come in this sequence. So now, the first base or codon, the first codon to be read here by the ribosomes, it's AUG. AUG with regard to eukaryotic cell. So in eukaryotic cell, the first base, the first codon to be read is this AUG. AUG is said to be initiating or the start codon. Initiating or the start codon for the fact that it is the first codon to be read by the ribosomes. The start codon. So this AUG specifies for an amino acid methionine. And this does not change. This does not change. It is always the first amino acid to be read as far as eukaryotic cells are concerned. Now, what about if it is in What about if it is in a prokaryotic cell? What is the initiating codon? So in prokaryotic cell, it is not, it is a modified methionine. So in prokaryotic cell, the start codon is N for mu methionine. For mu methionine. So this is the start codon with regard to prokaryotic cell. Prokaryotic cells. Okay, so that's what we have to know, the differences between prokaryotic cell and eukaryotic cell. But overall, the process of translation is nearly the same in prokaryotic cell and eukaryotic cell. So those are some of the exceptions which we are mentioning there. So now we said we have P site, E site, and S site. What happens next? In this case, we said that let's use eukaryotic cell. So in eukaryotic cell, the first codon here, we said is going to be AUG, that is methionine. Once this AUG is read, then now we said also that each codon will specify one amino acid. So it will read here AUG. Then this AUG is specified for an amino acid methionine. Then now, a particular tRNA which has got anticodons to this AUG has to be sent into the cytoplasm to go and pick this methionine and bring it here into the ribosomes. That's where now those processes aminoacylation are going to take place. So here now there's going to be this tRNA. So this tRNA is now going to be sent into the cytoplasm to go and pick that target amino acid. In this case, it is methionine. So we said that this tRNA has to have complementary bases to AUG. So complementary bases to AUG, we know A will form a complementary base with U, then U here A, then G, C. So these bases which are found on this tRNA are what we call the anticodon. And that is the reason why this arm is termed as anticodon arm, because it carries anticodons. Whereas these bases which are read on this mRNA are called codons. So codons are found on the mRNA, whereas anticodons are only found on the tRNA. So in this case, we are saying that AUG has been read by the ribosome there. Then a tRNA which has got anticodon to AUG is going to be sent to go into the cytoplasm and pick that amino acid which has got these anticodons. So in this case, that amino acid is methionine. So this methionine now, we said it is in its inactive form. All the 20 amino acids will exist in their inactive form. So what will happen here now? This mRNA has now to be first activated. The process of aminoacylation has begun. So it has to be activated. After being activated, it has to be charged. Once it's charged, it will now have to go and attach itself, we said, at the 3' end there of this tRNA. So this now is going to be the charged amino acid attach itself at the 3' end of that tRNA. So this molecule now, this tRNA, because it is carrying that charged tRNA or amino acid, is now said to be a charged tRNA. That is now termed as a charged tRNA. Now, just as the term suggests here, this is a transfer RNA. So now it's going to transfer this amino acid methionine to go where? Into the ribosomes. So this one, the first tRNA, which carries an amino acid methionine, this one will go straight into the P site. It is only this first amino acid which goes straight into the P site or the ribosome. That is the P site. So it will come here in the P site and deliver that amino acid. So what do we see at this point? What we see now is we have this tRNA, I'll present it like that, then with its amino acid there, like that. So the codon which was read here was AUG. So this tRNA is going to form those complementary hydrogen bonds with these anticodons or the anticodon at the anticodon arm or this Trna. So they are going to form that loosely complementary hydrogen bond. So just like that. I was supposed to show these. Let me show them here. A, U, G. Then that Trna has got those complementary bases U here, then A here, C there. Those hydrogen bonds. That's what is going to be formed there. Then this one has derivated now. Once it has derivated its amino acid, it then now becomes useless. So it has to leave the ribosomes. It will only leave the ribosomes through we said the E site. So E we said for exit. So it's now going to move this tRNA is going to move into the E site so that it can now leave the ribosomes like that. But overall, this amino acid will remain there in the P site. So let's get rid of this. So what we have at this point now, we have that amino acid in the P site. That's methionine, the first amino acid. Then this tRNA has left the ribosomes. So it's somewhere there. Once it leaves the amino acid there, it will now become what? Uncharged again. So that's exit. It will now become uncharged. Then, okay, at this point now, what will happen? We saw that the first codon to be read was AUG. Then we are done with AUG. The next step now is going to be the incoming codon. So the incoming codon has to be the next 3 bases. So the next 3 bases there, let's say we have CCA. Let's say we have CCA. This is what now is going to be read by the ribosomes. Once it's read by the ribosomes, a tRNA which has got anticodon to CCA is now going to be sent to go into the cytoplasm to go and pick this amino acid which is specified by that codon. So before this happens, there's going to be what is known as translocation. Translocation is going to take place here. So translocation, the movement of these ribosomes. So when they are done with the first codon, then now they are going to move 1 step ahead. So when they move 1 step ahead, this incoming codon is going to find itself here, where there was this AUG. Then AUG will be somewhere there. Then this is what now is going to be read there. So once that is read by this ribosome, then that particular tRNA which has got anticodon to that is going to be sent into the cytoplasm to go and pick that amino acid. So in this case, our target amino acid is that one. So let's say this is arginine. Then arginine now is what is now going to be activated. So arginine in the cytoplasm will become activated by being provided with ATP. Once it's activated, then it will be charged and attach itself at the 3' end of this tRNA. So let's say we have now that amino acid there at the 3' end there of this tRNA. So this tRNA, we know that it has to have anticodon that is now going to be GU. But as we know, Ubo hypothesis does provide that this position, the third position, can even mismatch. So even this one, on the first and second, these are the only positions we should not base mismatch. But the third one can mismatch. So this one can even have adenine there. That is accepted with Ubo hypothesis. Like that. Then this now tRNA. This tRNA now is also going to come where? Into the ribosome. So remember, this is the second tRNA to go into the ribosomes here. So this tRNA, second up to the last, these now will all enter the ribosomes through the A site. So this tRNA will come here, not into the A site, but it will come into the P site, but it will come into the A site here with its amino acids. So with its amino acid there, then it will enter the ribosomes through the A site. So all the incoming amino acids now will be entering the ribosomes through the A site. It is only the first amino acid that goes straight into the P site. that is methionine with regard to eukaryotic cell, and for me methionine with regard to prokaryotic cells. This is what now happens. So we have this tRNA in the A site. So we know that now the polypeptide chain will takes place in the P site. So there's need to transfer this amino acid from the A site to where? To the P site, so that it can come here and form that polypeptide bond. So what happens here now, there's need for another enzyme which can transfer that amino acid from the P site to the A site, sorry, to the P site where the polypeptide chain is growing. So that enzyme which transfers amino acid is what is known as Peptidotransferase, or Peptidotransferase. This is the enzyme which transfers amino acid from the P site, or from the A site to the P site. So it will transfer that amino acid and take it where? To the P site. Here they will be linked through, that is the peptide bond. So this will be the peptide bond, like that. So that is what will happen. Then when this happens, the next step now we see is that this tRNA which was carrying this amino acid, it will become useless, so it will have to leave the ribosomes. So it will leave it leaves the ribosomes through the E site. So I didn't mean to. Let's do this. Okay, we had this as our second bond amino acid, like that. Then this mRNA, this transfer RNA now is no longer here. So that transfer RNA now will find itself somewhere there. So it will leave the ribosome through the exit site there, like that. Then when this happens, we know this now has been read, this codon. Then there will be incoming, the next incoming codon is what now is going to be read. So let's say the incoming codon is now ACA. Translocation as well is going to take place where those ribosomes are going to move in this direction. Then this incoming codon is going to find itself in the P site there, somewhere there, so that when tRNA delivers the amino acid, it can form those complementary bases with these codon on the mRNA. So then another tRNA is going to be given an assignment to go and pick an amino acid which is specified by this codon, ACA. So let's say this amino acid is isoleucine. Then that tRNA is going to be given this assignment to go there and pick that leucine from the cytoplasm. So if we have all those processes of aminoacylation are going to take place, it will bind itself to the P site there, or to the 3' end of this tRNA. Then this tRNA, we said that all the incoming tRNAs will enter the ribosomes through the A site, like that. So this tRNA is also going to find itself in the A site there with its amino acid, like that. Then those anticodons will form complementary bonds with this codon which has just been read. That is what will happen. Then peptidotransferase is now going to transfer this amino acid from the A site to the P site, because it is in the P site where the polypeptide chain grows, just like that, linked through polypeptide bonds there. So that is now going to be our third amino acid, just like this. So this will continue. It will continue, then this chain will keep on growing, just like that. So this chain is what we call a polypeptide chain, linked through peptide bonds, just like that. This now, what has been taking place here, this is now the second stage in the process of translation, which is, we said, is elongation. So to elongate simply means to build. So this is what happens. So this process will keep on repeating just like that, not until it reaches what is now known as the termination site, where the process of translation has to come to an end. So it will reach the termination site. So what happens now here during termination? One characteristic of this termination is that, one characteristic of this termination site is that one of the three stop codon is going to be read by these ribosomes. So when it is read, so we know that we only have three stop codons. So we have UAG, we have UAA, then also have UGA. These are the only three stop codons which we have from the possible 64. These do not code for anything. As a result, they are termed as stop codons. Stop or terminating codons. That's one and the same, like this. So we are saying that one of these codons is going to be read by these ribosomes. So let's say it is UAG. UAG will be read by the ribosomes. Then there's no tRNA in the cytoplasm which has got anticodons to UAG. Now, because of this is now going to influence a particular molecule which is known as a release factor. Once one of the stop codon is read or comes across in the ribosomes, then a molecule known as a release factor is going to be activated. A release factor is going to be activated and enter this ribosome through the A site. So that release factor will enter the ribosomes through the A site here, just like that. It's very much similar to the amino acids, but just that it does not specify for an amino acid. So once it gets there into the P site, then now going to move into the P site, then join itself to this growing polypeptide chain. Once it joins there, then it's going to release this growing polypeptide chain. This growing polypeptide chain is now going to be released, and this is the reason why it is known as the release factor, which is abbreviated as RF. So there are different types of release factor. There is release factor 1, factor 2, and factor 3. So the function, let me maybe talk of the function of release factor 3. The function of release factor 3 is to activate release factor 1 and factor 2. Then this release factor 1, it reads the stop codon UGA and UAA. Then these are read by release factor 1, if I'm not mistaken. Then the remaining one, which is UAG, this one is read by release factor 2. Then we have release factor 3, which its function is just to activate release factor 1 and release factor 2. That's what happens during the process of translation. So at this point, what we see that the process of translation has come to an end because this growing polypeptide chain now has been released. So what do we see? What will happen with this assemblage? What will happen now to this polypeptide chain as well as to this complex mRNA, the ribosomes, and everything? What will happen next after the process of translation has come to an end? What will happen at this point now? You learned about organelles of the cell. We have one special organelle of the cell which is Golgi apparatus. So we said that Golgi apparatus, the function of Golgi apparatus is for protein modification. So this is where now this growing this polypeptide chain which has been released from the ribosome is going to go. It's going to be transported and go straight where? To this Golgi apparatus, where it can be modified structurally and chemically. So come to this later on, what happens in the Golgi apparatus. Then first now let's talk of what will happen to this complex now. What will now happen to the ribosomes as well as this mRNA. So when translation comes to an end, that growing polypeptide has been released. This now, the large and the small subunit are going to dissociate. They are going to dissociate, that is, this complex will no longer be in existence. Then this mRNA now is going to be degraded or broken down. It's going to be degraded or broken down. And kept by the cell so that it can be reused in the second translation process. So this is what will happen. So what we'll see is this large subunit is going to find itself away from the small subunit. Then the mRNA will also find itself on its own. That is now the process of translation has come to an end. So at this point, what we have now, we only have these base sequences of amino acid sequences. So we looked at proteins. We said that we have different types of proteins: primary, secondary, tertiary, as well as quaternary. So this, if these are amino acids, this is the polypeptide chain. This type of protein, the way it is here, it has not been modified. It has just been released. This is an example of a primary protein. A primary protein, just like this. So as we know, the function of each and every protein depends on its structure. We have secondary proteins, primary, tertiary, and quaternary. So this now meaning that if this protein produced here, let's say that it is hemoglobin. Hemoglobin is a quaternary protein, so it has to be modified to a state where it fits to be a quaternary protein. So that's why this polypeptide chain, which has just been released, it has now to go where? To the Golgi apparatus. That's where it's going to be. That's where modification takes place. So this now this process that we are explaining now, this is what now we call post-translational modification. So just as we have post-transcription modification, even here we also do have what is known as post-translational modification. So we have post-translational modification. What happens after translation? This is what happens now. So there are two types of post-translational modification. The first one is chemical modification, and the second one is structural modification. So we are saying we have two types of these post-translational modification. The first one is structural modification. So structural modification has to deal with the modification of the structure. Then we have the second type of post-translational modification, which is chemical modification. So chemical modification does involve addition of chemicals to that particular protein. So we know that there are certain proteins which cannot function without an addition of particular elements, such as hemoglobin. This element, iron, has to be added. So that is an example of chemical modification in post-translation. So firstly, when we talk of structural modification, this has to deal with modifying the structure. So modifying the structure to become one of the four types of proteins, which is primary, then secondary, or tertiary, or quaternary. So this modifying to become one of these, that is what is known as structural modification, and this is done by Golgi in the Golgi apparatus. So once it is modified structurally, then that particular protein can now be released and go and released so that it can be used with its function, to carry out its function in the cell. Then there are some proteins which cannot function just when they are just been modified structurally. A chemical has to be added. Those are now what we call chemical modification. So we have different examples of chemical modification. Different chemicals can be added to different proteins with regard to different functions that different proteins perform. So there is what we call the first, we can talk of protein phosphorylation. Protein phosphorylation is one of the examples of chemical modification. So in this case, meaning that a phosphate group has to be added to that particular protein. So here involves addition of a phosphate group. in phosphorylation. Then we can also talk of methylation. So methylation has to deal with the addition of methyl group to that particular protein. So addition of methyl group is added to that particular protein. Hence the process becomes methylation. We can talk of different examples. Glycosylation. This is I think the one which you have come across with when you are looking on cells. I think you came across such. So glycosylation, this one now involves the addition of carbohydrates. So this one involves addition of carbohydrates to a protein. Carbohydrates we know are sugars. So glycoproteins, they are found on the surface of the cell membrane of a cell. Their function mainly is for cell-to-cell recognition. For one cell to be able to recognize the next cell that this is my fellow type of a cell. Proteins or cells do contain these type of proteins, glycoproteins. So the process by which a carbohydrate is added to that protein is known as glycosylation, whereas a protein is what is called glycoprotein. We also have lipoprotein. So lipoprotein, the process here has to be, okay, we have lipoprotein. So this one has to involve the addition of a lipid. That protein comes to be called a lipoprotein. So you can talk of different types of molecules that can be added to this protein. Then that particular protein now can perform its normal function. So we have a lot, but these are some which I can list. So this is what happens actually in post-translational modification. This is all what happens in general with regard to the process of translation. Then finally we are going to have these proteins which will give us which will now form different molecules in the body that makes it possible for bodies or these living organisms to carry out their functions. Alright, so this is what we had for you in this video. Make sure that you look out for some more videos for these other topics which come ahead. Thank you for watching.